the important classes of inhibitors, which is your folate pathway. Now, folate pathway, I am discussing it right now. We'll be using it in the next chapter as well, which is your antimycobacterial. You will be also studying this in the class of anti-infectives. So as a pathway, the pathway will remain same, but with respect to the organism, the enzyme will differ, which means I will have the same architecture of the pathway. So I'll have same enzymes. So I'll also have dihydrofolate reductase present in Plasmodium, which we'll study today. And I'll also have dihydrofolate reductase in Mycobacteria, which will act as antituberculosis agent. I will also have DHFR as an enzyme present in humans, and if I am trying to inhibit that, I'll have anti-cancer agent. So the pathway will remain same. Name of the enzyme will also remain same, but what will differ is the three-dimensional structure of the enzyme. And since the enzyme components change, thereby you achieve what is known as selectivity, where we are targeting only parasite, we are not affecting the dihydrofolate reductase in humans. So for folate pathway, the pathway begins right from the first step, where we call it as de novo pathway, because bacteria can take up para-aminobenzoic acid and synthesize their own dihydroteroic acid. And this is catalyzed by the enzyme dihydroteroid synthase. And all your sulfonamides, your sulfa drugs, inhibit this dihydroteroid synthase. And this is not there in human at all. So there is no issue of selectivity. That pathway or this enzyme is selective to bacteria only. Now when this dihydroteroic acid takes up glutamate, it forms dihydrofolate. So this is what bacteria is doing. So from where do humans get this dihydrofolate? They get it from diet. So your dihydrofolic acid, humans get it from their diet, and then it is converted to tetrahydrofolic acid. And for conversion of DHF, this is abbreviated as DHF, dihydrofolic acid or dihydrofolate, to THF, tetrahydrofolate, the key enzyme is dihydrofolate reductase, and that is inhibited by all your agents here, trimethoprim and pyrimethamine. So if you are inhibiting the bacterial enzyme, you have drugs like your trimethoprim, pyrimethamine. If you're inhibiting human DHFR, you have drugs like your methotrexate, which affects human DHFR. And this tetrahydrofolate, it is important because this tetrahydrofolate, through further transformations, is involved in one carbon transfer, and whereby that species, whatever is generated further, it takes part in synthesis of DNA, RNA. It is also involved in synthesis of proteins. It is also involved in thymidine, and thereby it is also involved in the process of DNA through the thymidylate pathway. So all these taken together, you will realize that this conversion of hydrofolate to tetrahydrofolate is essential for cell survival. It is essential for cell replication. So if this is not provided to the organism, or humans, if we are talking about anti-cancer agent, you will not have cell replicating, you will not have cell surviving, which causes the cell to die. And this cycle, what is observed is tetrahydrofolate, once it transfers the methyl, it is reduced back again, it is oxidized back again to dihydrofolate, and this cycle continues. So it is not that THF is lost, it is converted back to dihydrofolate, and this process keeps continuing. Now what is also observed is, supposingly you give only trimethoprim, the bacteria has a smarter way of devising its own methods. So what is done is, you don't inhibit just one enzyme, inhibit both the enzymes. So dihydroteroid itself, it is inhibited, so dihydroteroic acid is not formed, and ahead of it you are also inhibiting your DHFR. So one pathway or one or two enzymes before this you are inhibiting, and then given together, what will happen is both the enzymes are acting in the same pathway, and you see a overall inhibition, and the cell cannot be rescued. What is normally seen is, bacteria are very smart. If you try and inhibit one process, they try and identify a rescue process and they still survive. But if you are inhibiting two enzymes of the same pathway, then there are hardly any chances that the organism will survive. So this is your first understanding of what is known as combination therapy, whereby in the combination therapy, you are giving one sulfa drug plus one DHFR inhibitor. That is normally done for your anti-infective therapy. And same way, supposing you are having, if you are planning anti-cancer therapy, methotrexate is inhibiting DHFR, and you have 5-fluorouracil or flucytosine, which are inhibiting the thymidylate process. Given in combination, it will be a very severe anti-cancer agent. You can adjust the dose obviously, but then you can plan of giving them together so that you prevent the rescue process. So this is the summary of your folate pathway, and what we'll be studying is agents which are used as anti-malarial specifically, not antibacterial in general. So for your syllabus, the first agent is pyrimethamine, and pyrimethamine as a DHFR inhibitor is a very potent DHFR inhibitor. Look at its affinity. What is said is, with respect to the host enzyme, it is 1000 times more selective for the Plasmodium species, which means it is going to inhibit Plasmodium 1000 times more than the human counterpart of the enzyme, which gives it a lot of selectivity to treat plasmodial infection. Supposingly, imagine I say that my drug has 1:10 selectivity, which means if it is inhibiting human at 1 microgram, 10 times of the concentration is required to inhibit the other form. Vice versa, if I tell you that at concentration 5 milligram per ml, the drug is inhibiting malarial parasite, but for human it is inhibiting at 10 mg per ml, which means hardly any difference, and thereby selectivity is very less. And when I say selectivity is very less, if the DHFR in human is also inhibited, you are trying to treat the patient for malaria, but you will kill the patient by causing unwanted toxicity to the cell. So that is why whenever you have enzymes present in both organisms and the host, you have to have a very high level of selectivity. We'll be studying the selectivity as a concept in other chapters as well. So please remember this concept of selectivity. Layman's term, you have two enzymes, you have to inhibit only the organism. Nothing should happen to the host enzyme. That is the concept of selectivity. Now for this agent, what is normally done is it is given as a combination. Fansidar is the combination where pyrimethamine is combined with a long-acting sulfonamide, and that example of long-acting sulfonamide is sulfadoxine. So sulfadoxine along with pyrimethamine forms the combination drug which is Fansidar. And the advantage obviously is entire folate pathway will be inhibited. So you are disrupting the folic acid synthesis in the plasmodium if you are inhibiting both of them. Now same way, pyrimethamine is also given in combination with quinine to treat or to prevent, if you say a chloroquine resistant malaria has been observed, you cannot give chloroquine, you need some other drug. Quinine you cannot administer in large form. So what is done is you give quinine. Now you are giving it in combination, so the dose will get reduced, and quinine and pyrimethamine together can now act on resistant species. And the reason why this becomes important is many plasmodial species eventually have become chloroquine resistant. And chloroquine was one of the best drugs because it was able to kill various forms of the plasmodium species. And therefore, when you're trying to develop a drug, if something is useful for chloroquine resistant forms, it has more value than anything else. Now, for pyrimethamine, one of the important methods by which the organisms develop resistance is point mutation in any of these enzymes. So when I say point mutation, for now just remember your gene which is preparing this protein, in that gene a mutation has taken place and now this DHFR has changed. Since the DHFR has changed, the drug is no more able to bind DHFR and therefore loss of activity. The same way, if there is a DHPS is dihydropteroid synthase. If there is mutation in the gene which makes this protein, you have a different protein generated now, and your sulfadoxine will not act on DHPS. Therefore, you will have overall resistance. The drug will become ineffective and the organism will still survive. So this is for your pyrimethamine. We have one more drug to study, which is proguanil. Now proguanil, the name itself is saying proguanil. It's a prodrug of the actual drug, which is cycloguanil. And this was the example I was talking about, where the initial form or the proguanil is inactive. Only when it undergoes metabolism, you have the active metabolite or the species which is actually contributing to the activity. And your cytochrome enzyme CYP2C19 catalyzes this conversion. So proguanil on metabolism forms the active cycloguanil. So what is observed is, whenever there is some population which is lacking this enzyme or which is having what we can say is lesser active enzyme, which is known as polymorphic enzyme forms, they are not able to convert proguanil into active guanil. So proguanil not converted to active cycloguanil, and therefore you will have no effect, which is your example of genetic modification, which happens due to polymorphic variation. So certain population, you have to identify whether that population is receptive to this metabolism, which is done in genotyping studies, where you check whether this enzyme is normal or it is not normal. And if the enzyme is normal, only then the drug will be active. And since it is, again, we are studying it in the folate pathway, it's easy for you to now remember that it is going to affect DHFR, and therefore it interferes with deoxythymidylate synthesis, which means overall it is then interfering the DNA RNA synthesis and your protein synthesis. Cell dies. And what is again observed here is, due to mutation, there are amino acid changes which are seen near the active site. So for now, remember, as a protein, the drug goes and occupies a specific location on the protein, and only that part of the protein becomes important for us, and that is known as active site. Now in the active site, there are certain amino acid which take part in what we can say is binding of the drug to the active site. So they are going to act as agents which will hold the drug in place. Now imagine these amino acids, if they are changed, the drug it is no more interested in binding to the protein, which loses its activity towards the protein. So such amino acid changes near the binding site causes loss of activity, which is the primary reason for development of resistance. Now for proguanil, this is your guanidine type of group. Guanidine is when instead of urea, in urea you have NH, that O also is N, gives you guanidine. Since it is N, it becomes NH. So it's that guanidine type of analog where you have, if you closely look, NH becomes the common thing for two guanidines. And inside what happens is, it cyclizes and you get cycloguanil. So looking at the mechanism closer, when oxidation is taking place, you have this cycloguanil. It is the same structure represented in the way so that the cyclization can be easily understood. So you have C double bond NH, NH, C double bond NH. Now your terminal C gets hydroxylated. So that hydroxylation is followed by an internal, in simple terms, it is internal loss of water molecule. So H and OH lost, whereby you get this double bond, and the carbon becomes electron deficient, which is then attacked by the adjacent electrons from N. And therefore you get this cyclization taking place, and you generate this initial form and tautomerism, because once the tautomerism is taking place, once the bond migrates, what you generate is a conjugation, and this conjugation therefore becomes more stable compared to this exocyclic double bond. This becomes endocyclic conjugated 1,4-butadiene kind of 1,3-butadiene kind of system, which makes it more stable compared to this earlier tautomeric form. So this is for your practice, you can remember it as this N is directly attacking the carbon, and since it is directly attacking the carbon through this intermediate, it is forming a cyclic structure, which is your cycloguanyl, and that can happen only when the first step of oxidation takes place. Rest of the things are spontaneous things happening inside the body, but this first step where OH is getting attached, that is crucial for us. Only then the further cyclization can take place. So this is your proguanyl